Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Assalamu alaikum. Today I will discuss my topic name is FTIR spectroscopy. Topic to be discussed: introduction, theory of the Fourier transform, infrared spectroscopy, instrumentation of FTIR, advantages, disadvantages, and applications. Firstly, we discuss the introduction. Fourier transform infrared spectroscopy is a sophisticated analytical technique used to study how molecules interact with infrared light by measuring how samples absorb infrared radiation at specific frequencies. FTIR spectroscopy provides a valuable insight into the molecular structure and composition of the materials. This technique is widely employed in chemistry, pharmaceutical and material sciences, and environmental analysis due to its ability to identify the functional group, determine the molecular structure, quantify the substances in a non-destructive manner. FTIR spectroscopy is preferred over the dispersive method of IR spectral analysis for the several reasons. It is a non-destructive technique. It provides a precise measurement method which requires no external calibration. It can increase scan speed, collecting a scan every second. It is mechanically simple with only one moving part. Theory of the FTIR. Fourier transform infrared spectroscopy was developed in order to overcome the limitations encountered with the dispersive instruments. The main difficulty was the slow scanning process, a method for measuring all of the infrared frequencies simultaneously rather than individually was needed. Solution was developed which employed a very simple optical device called an interferometer. The interferometer produces a unique type of signal which has all of the infrared frequencies encoded into it. Most interferometers employ a beam splitter which takes incoming infrared beam and divides it into the two optical beams. One beam reflects off a flat mirror which is fixed in place. The other beam reflects off a flat mirror which is on mechanism which allows this mirror to move a very short distance, typical a few millimeters away from the beam splitter. Two beams reflect off their respective mirrors and are recombined when they meet back at the beam splitter. Because the path that one beam travels in a fixed length, other is constantly changing as its mirror moves. Signals which exhibit the interferometer is the result of these two beams interfering with each other. The resulting signal is called an interferogram, which has a unique property that every data point function of the moving mirror position which make up the signal has information about every infrared frequency which come from the source. Because the analyst requires the frequency spectrum plot of intensity at each individual frequency. In order to make identification, the measured interferogram signal cannot be interpreted directly. Hence, Fourier transformation is performed by computer, which then present the user with desired spectral information for the analysis. The next, I discuss the instrumentation of the FTIR spectroscopy. FTIR spectroscopy rely on the specialized instrumentation designed to analyze the interaction between the infrared light and material. Here's overview of the key components and principal FTIR instrumentation. Infrared light source. FTIR spectrometer utilizes a source that emits a broad spectrum of infrared light, typically from 2.5 to 25 micrometers. Conversion spectrum, the mid-infrared region where the most molecular vibrations occur. Interferometer. The core of the FTIR spectrometer is the interferometer, commonly a Michelson interferometer. It consists of the beam splitter, divide the incoming infrared beam into the two parts. Fixed mirror, the second one is moving mirror. One path reflects off a fixed mirror, the other path reflects off a mirror that moves back and forth. The interferometer creates the interferograms, which is time domain signal representing the intensity of the infrared light as a function of the path difference between the two beams. Sample compartment. This is where the sample under investigation is placed. The infrared light passes through a reflector of the sample, causing a specific wavelength to be absorbed based on the sample molecular composition. Detector measures the intensity of infrared light after it interacts with the sample. These are the diagram for the FTIR spectroscopy. Advantages of FTIR. Fourier transform infrared spectroscopy is a powerful analytical technique used to identify the organic, polymeric, and in some cases inorganic material. Here are some key advantages. FTIR. Speed. FTIR can quickly collect the data over the wide spectral range. The use of Fourier transform allows for rapid acquisition for spectral data compared to the traditional dispersive infrared instruments. Sensitivity. FTIR is highly sensitive and can detect even small amount of the substances. It is capable of identifying the functional groups and chemical bonds with high pre. Non-destructive. FTIR analysis is generally non-destructive, meaning sample can be preserved for the further testing or use. Quantitative analysis. FTIR can be used for the both qualitative and quantitative analysis. The intensity of absorption bands is proportional to the concentration of the sample, for the accurate quantification. Versatility. FTIR is versatile and can be analyzed wide range of the sample, including the solid, liquid, and gases. It is commonly used in various fields such as chemistry, material sciences, environmental sciences, and pharmaceuticals. Minimal sample preparations. FTIR requires a minimal sample preparations which save times and reduce the potential for the contaminations and sample alteration. Wide range of applications. FTIR can be used in numerous applications including quality control, fringing analysis, environmental monitoring, and material identification. High resolution. FTIR provides the high spectral resolution which enable the detailed analysis of the complex mixture and identification of subtle differences in molecular structure. These advantages make FTIR a valuable tool for the scientist and researcher in many fields. Disadvantages of FTIR. While the FTIR spectroscopy is a powerful analytical technique, it does have some limitations and disadvantages. Limited to IR active molecules. FTIR is only effective for the molecule that have the change in dipole moment during the vibration. Molecule that are IR inactive, such as homonuclear diatomic molecule like O2 and 2, cannot be detected. Water interference. Water vapor and other atmospheric gases can absorb the infrared radiations, leading to interference in the spectra. This can be problematic, especially for the sample that are sensitive to the moisture. Sample preparation. Although FTI generally require minimal sample preparation, certain samples might need a specific preparation techniques that can be time consuming. Surface sensitivity. FTIR may not be suitable for the surface analysis of the material as it typically provides the information about the bulk composition. Surface sensitive techniques like X-rays, photoelectron spectroscopies, or secondary mass spectrometry might be required for detailed surface analysis. Quantitative limitations. While FTIR can be used for quantitative analysis, it requires the calibration with known standards. The accuracy of quantitative results can be affected by the sample homogeneity and the preparation methods. Complex spectra interpretations. Interpretation of FTIR spectra can be challenging, especially for the complex mixture. Overlapping absorption bands can be complicated the identification quantification of the specific components. Penetration depth. For the solid sample, penetration depth of infrared radiations can be limited, potentially leading to incomplete analysis of thicker and multi-layered samples. Cost high. High quality FTIR instrument and their maintenance can be expensive, Additionally, specialized accessories and software for the specific application can add to the overall cost. Resolution limits. While the FTI provides a good resolution, it may not always be sufficient for the highly complex mixture or distinguishing between the very similar compounds. Environmental sensitivity. The process of carbon dioxide and other atmospheric contaminants can be introduced noise or interfere with the measurements necessitating the use of the merging system or other methods to mitigate these effects. Applications. FTIR spectroscopy a wide range of applications across the various fields due to its ability to identify quantify different chemical compounds. Here are some key applications: chemical analysis, pharmaceutical and environmental sciences. Firstly, chemical analysis. Identification of the organic compound. FTIR is commonly used to identify the organic compounds by analyzing their functional group. Polymer analysis. It is used to identify and characterize the polymer and their additives such as plasticizers and stabilizers. Pharmaceuticals. Drug analysis, quality control. FTIR is used to identify active pharmaceutical ingredients and excipients in the drugs. It ensures the consistency and quality of pharmaceutical product by detecting purities and degradations product. Environmental sciences, air quality filtering. FTIR can detect the pollutants and gases in atmosphere such as carbon dioxide, carbon monoxide, NO and SO2s. These are gases. Water quality analysis. It is used to analyze the contaminants and organic compounds in water sources. Forensic sciences. Substances identification. FTIR helps in identifying the unknown substances found in crime scenes, such as drugs, explosives, and fibers. Documentary analysis. It can be analyzed the inks and proper paper to detect the forgery and document alterations. Material science is characterization of material after a year is used to study the composition properties of the material, including the ceramics, glasses, and metals. Surface analysis, it helps analyzing the surface treatment and coating. Food and agriculture, food quality control. FTI can detect the alterant and contaminant in food product, ensuring their safety and quality. It helps nutritional analysis, it helps in determining the nutritional content and composition of the food items. Industrial application process monitoring. FTIR is used in industrial process to monitor the chemical reactions and ensure the product quality. Quality assurance, it ensures the consistency and quality of raw materials and finish the product to various industry. These diverse applications highlight the versatility and importance of FTIR spectroscopy and scientific research industry and various pharmaceutical fields. Thanks for watching.